So basically, as you were saying, you end up with fragments of every possible length of the exon. So what we do next is then we load the fragments onto the gel and apply an electric current to separate the fragments by weight, as the smaller or lighter currents move quicker than the larger one. So I'm going to show you quickly what it would look like. So here we're loading on all the different exon lengths onto the gel. We're then applying the current where the smaller gels will move quicker, or the smaller or the lighter gels will move quicker through the gel, and therefore they'll get tagged by the laser first, and all the gels will move through. And as they get tagged by the laser, they get recognized for the probe color, and that creates a pattern. So each nucleotide has a different color probe attached to it. So as you can see there, G, T, A, C. They all have different colored probes, so you start forming your sequence of DNA. So basically, at the bottom of the gel we have the laser, which detects the fluorescent tag, so we should build up our sequence. And this enables us to diagnose at a molecular level by comparing the sequence of the patient's DNA to normal DNA. So some summary of the Sanger sequence: it uses PCR to amplify genes of interest. It looks at each base, identifies as small substitutions or indels, which is the posh term for insertion or deletion of bases. However, you need to know what you're looking for. You need to know what abnormality or what indel you're looking for. We move on to next generation sequencing now. It's targeted, and it allows us to sequence the whole exome or genome rather than going into a gene already in mind. So the summary of next generation sequencing: it looks at each base, identifies substitutions or indels, can look at all the genes at once, and it has a different chemistry from Sanger sequencing. Now I'm just going to show you a quick summary table. So basically that's how, oh my god, that will basically be the probe that you get. All the probes will be identified and it will form a pattern, and that is the sequence of the 4 nucleotides. I'm going to show you a summary table now. So you can see the test, the structural abnormalities, the DNA level, and the prior knowledge needed. So G-banding, it shows chromosome number, translocations, and large deletions or duplications. The DNA level, well, it's looking at like the macro structure of DNA, because it's looking at chromosomes. And prior knowledge needed, none. Fish chromosome, its structural abnormalities, it looks at its chromosome number, translocations and deletions and duplications, but around 5 megabases. DNA level, as before, is looking at the macro structure, and prior knowledge needed, some or none. QFPCR, it looks at the chromosome number, and QFPCR, looking at the chromosome number, how it does that was through the what are these called again? Microsatellites, as we were saying. And then the array CGH looks at micro deletions, micro duplications, and CMVs, and looks at these basically through CMVs. DNA level, don't really need to know, and prior knowledge needed is none. Sanger sequencing, it's looking at like micro abnormalities in bases, so it's looking for substitutions and small indels, and you need to know the suspected genes where these abnormalities may occur. And next generation sequencing is exactly the same. It looks at substitutions and small indels. However, there's no prior knowledge that is needed. Oh, thank God.